Here to talk about nucleic acids. Nucleic acids are biological polymers, generally composed of 4 different monomer units, termed nucleotides. The unit length of one molecule can be as much as a billion nucleotides. Although the biological roles that nucleic acids fulfill are diverse, the structure and function of each molecule is elaborated from a common set of general properties.
Okay, now we will talk about the structure of nucleic acids. Nucleic acids contain a nucleotide, which is a monomer that produces nucleic acids. It contains 3 major components: a 5-carbon monosaccharide sugar, a nitrogenous base, and a phosphate group. Nucleotides are bonded together from sugar to phosphate of opposing nucleotides. Thus, the sugar of a nucleotide is bonded to the nitrogen of the other nucleotide. Bonds are held intertwined by a molecular force. And this is a picture of the structure of a nucleic acid, or nucleotide. The phosphate group, which is bonded to the sugar, and the sugar bonded to nitrogenous bases. And these are the multiple nitrogenous bases that are contained in nucleic acids. Alright, now we'll talk about the class of nucleotides. There are 3 different examples of class of nucleic acids. One is DNA. DNA is deoxyribonucleic acid. It is found in living cells. The structure of DNA contains 2 extended chains of nucleotides twined together in the formation of a spiral staircase. This spiral staircase is known as a double helix. It consists of a phosphate group as well, 5-carbon deoxyribose sugar, and a nitrogenous base. The nitrogenous bases are on the inside of the double helix. The nitrogenous bases contain adenine, thymine, guanine, and cytosine. These are known as complementary. Thymine and adenine are bonded together. Cytosine and guanine are bonded together. These bondings are known as complementary base pairing. The main function of DNA is to store genetic material. Next is RNA, ribonucleic acid. It differs from DNA. The structure of ribonucleic acid is made of adenine, cytosine, guanine, and uracil. This is different than DNA because thymine never appears in RNA. RNA is made up of a sugar ribose, which differs from DNA because DNA contains deoxyribose sugar. RNA has one single chain with no bonds between the nitrogenous bases in the nucleotide. RNA gives permission of cells to apply the material that appears in the DNA and is used to make proteins containing the order of amino acids that corresponds to the nitrogenous bases in the RNA. And finally, the last example of class is the peptide nucleic acid. The peptide nucleic acid is an artificial. It is abbreviated with PNA. It is different from RNA and DNA because it's made of glycine to form the peptide bonds instead of the deoxyribose and ribose sugars. This nucleic acid was formed in 1991 and is artificially made in the laboratory. This allows scientists to become more and more detailed because of their great bonding strength. This is a picture of DNA with a double helix and the base pairings in the middle. Bottom left is a picture of the RNA with the single bonds with the different uracil, adenine, cytosine, and guanine. And on the right is an example of a peptide bond.
Properties. They have a negative net charge due to the phosphate bonds. DNA does not usually exist as a single molecule, but instead as a pair of molecules that are held tightly together through a double helix. They are made up of nucleotides. The backbone of DNA strands is made up from alternating phosphate and sugar residues. DNA double helix is stabilized primarily by 2 forces: hydrogen bonds between nucleotides and base stacking interactions among aromatic nucleobases. As you can see, these are pictures of the DNA with the properties of the double helix, the phosphates, the bases, and the deoxyribose. Potential uses. In a study done by Oxford, they were able to take cells from the nose of a dog and put it into the spinal cord so that the paralyzed dog would be able to regenerate cells and be able to walk once again. Through the use of genetics, this was able to be accomplished. If further research is done, this kind of work could be done to even help humans who have been either paralyzed or have sort of dysfunctioning diseases such as cancer. Thank you for watching our project.